megakaryocyte. Megakaryocytes develop to the megakaryocyte. Megakaryocyte. Yeah, megakaryocyte. And this megakaryocyte is very, very large or huge loose cell. These megakaryocytes not able to pass through the sinusoidal capillary of blood vessels. Yeah, and that's why when the megakaryocyte, because it's very loose cell, try to pass through the intracellular space of sinusoidal capillary by this endothelial cell, or endothelial cells of sinusoidal capillary fragmented. That means fragmented to the small cytoplasmic fragment, which is known as platelets or thrombocytes. What is thrombocytes? What is platelets? It's cytoplasmic fragment of megakaryocytes. Yeah. This is the development sequence of the thrombopoiesis, or formation of platelets. Yeah. Formation of platelets called thrombopoiesis. Yeah. These platelets not present in the red bone marrow. Platelets circulate within our general circulation. Okay. This kidney also produce another hormone which called erythropoietin. Yeah. If the RBC or red blood cells decrease, kidney responsible to produce erythropoietin. Because if erythrocytes or red blood cells decrease, oxygen supply also decrease in our tissue. In response to the hypoxia, kidney produce erythropoietin. Erythropoietin also activate or stimulate the differentiation of myeloid progenitive cells. This myeloid progenitive cells under the influence of erythropoietin differentiate to the erythroblast. Excellent. Proerythroblast. Proerythroblast further develop, change to the basophilic erythroblast. Yeah. Basophilic erythroblast convert to the polychromatic erythroblast. Polychromatic to the orthochromatic erythroblast. Orthochromatic erythroblast develop or convert to the reticulocyte. Reticulocyte finally making our RBC. Yeah. This is the development sequence of RBC, which known as erythropoiesis. Yeah. Okay, one thing, one question. Okay, why called basophilic erythroblast polychromatic orthochromatic? Why?
Cytoplasm basophil.
Yes, of course. Why cytoplasmic basophilic in this cell?
Red color.
Because they're secretary.
Secretary.
This cells is very large or huge cells with loose nucleus. These cells at the first proerythroblast replicate the genetic material, make more RNA, DNA, because prepare for synthesis of a specific protein which called globin. Yeah, that's why. What do you think? Genetic material or RNA, DNA, it is acidic component of cell or basic component of cells?
Yes,
Acidic component of cells. That way, acidic component of cells stand by basic dyes. Yeah, if we see under the microscope here in the cytoplasm, because genetic material more RNA, ribosomes, and that's why stained by basic dyes, that's why called basophilic erythroblast. In the next stage, this basophilic cell convert to the polychromatic. Well, why? How you think? Why basophilic erythroblast convert to the polychromatic? Because now these cells make more RNA, replicate the genetic material, make more ribosomes actually, and now translate. It means make more protein. That word protein is this basic component which is stand by acidic dyes. In the cytoplasm, proteins stand by acidic globulin by acidic dyes, and genetic material ribosomes stand by basic dyes. That's why this is the polychromatic. Under microscope look polychromatic, colorful. Next stage, in the orthochromatic erythroblast, how look orthochromatic? Genetic material condensed. It means the basic component condense. Basic component condense. Yeah. And only acidic component condense. Basic component remain and stand by acidic dye. It means an orthochromatic erythroblast under the microscope look reddish because in the cytoplasm present globin, protein. Yeah. Now this cell changed to the reticulocyte. Reticulocytes, yeah, this is reticulocyte because having condensation of genetic material and nucleus should eject out. This is reticulocyte which contain sufficient globin. Yeah, enough globin. Here is back of globin. But these cells, reticulocytes, need for iron. Hey, to catch the iron and make hemoglobin. Yeah, hemoglobin. Now, then the next stage, reticulocytes change to the RBC. RBC is a biconcave cell. Here you can see biconcave cells which acidic peripheral part which is stained well and with pale center. Yeah, this is our RBC. Peripheral part stained well by acidic dyes and central part pale. Yeah, because presence of hemoglobin. Yeah, hemoglobin. This is the development sequence of our RBC and erythropoiesis. Okay, this is your first course topic. You should know hematopoiesis, formation of blood cells. Yeah, and your today's topic is histological structure of red bone marrow. It's so simple. You should draw and you should describe. Now, if myeloid progenitive cells stimulated by another factor like interleukin 1, interleukin 3, granulocyte colony stimulating factor, a granulocyte colony stimulating factor, myeloid progenitive cells differentiate to the myeloblast. Yeah, differentiate to the
Blast.
myeloblast. Myeloblast, F stimulating factor, granulocyte colony stimulating factor. Myeloblast develop or differentiate to the neutrophil, basophil, eosinophil, promyelocyte.
Myelocyte.
Promyelocyte. Promyelocyte. Neutrophil, basophil, eosinophil, promyelocyte. The promyelocyte further develop to the myelocyte. Neutrophil, basophil, eosinophil, myelocyte.
Myelocyte.
And then convert to the metamyelocyte.
Myelocyte.
Myelocyte. And then make our granulocytes. Granulocytes. Do you know granulocytes? Granulocytes is neutrophil, basophil, eosinophil.
Yes.
How look neutrophil? This is nucleus. This is the neutrophil. Multilobar or multi-segmental nucleus with acidic and basic granules. Here you can see the basic granules and also acidic granules. That why called neutrophils. The same amount of acidic and basic granules. Okay, this is neutrophil. Second granulocyte is basophil.
Only basic.
Yeah, basophil look like this. This is the H-shaped nucleus and only basic granules.
Granule.
Yeah, this is the
Basic granule.
basic. Next granulocyte is
Eosinophil.
Horseshoe shaped.
Is a bilobar nucleus,
Nucleus.
like food.
Head.
Okay, here you can see only the
Acid.
acidic granules.
Granules.
Acidophilic granules.
Acidophilic granule.
Acidophilic
Granule.
This is the granulocytes. Leukopoiesis, formation of granulocyte. And also, if the stimulating factor, a granulocyte colony stimulating factor, this myeloblast convert to the promonocyte.
Promonocyte.
Promonocyte to the
Monocyte.
monocyte. Yeah, monocyte it is a granulocyte. A granulocyte.
Site.
Here you can see like this. Yeah. This is the monocytes. Monocytes, if leak out from our blood vessel or left the general circulation and goes to the different tissue, convert to the macrophage, and also the name of them change. For example, if goes to the bone tissue called osteoclast.
Osteoclast.
Yeah, it goes to the epidermis called Langerhans cells of dendritic cells of Langerhans. It goes to the lung tissue called Das cell. It goes to the central nervous system called microglia. In the kidney called mesangial cell. All this in the liver called Kupffer cell.
Kupffer cell.
Yeah. All derived from monocyte of a Okay, about composition of blood, if you remember, in 1 microliter of blood, in 1 microliter of blood, 55% of our blood it is plasma.
Plasma.
Yeah, plasma, 92%, 92 to 93%
Water.
water, H2O.
H2O.
And about 7% it is plasma protein.
Plasma.
Plasma protein include albumin
Globulin.
and globulin and fibrinogen. Albumin is the most abundant, about 60%, here 37%, and here is 4%. Other on 1% of the plasma, it is it is nutrients, it is
Electrolytes.
respiratory gas, oxygen, carbon dioxide, amino acid, glucose, and vitamins actually, sodium, potassium, magnesium, chloride is the
1%.
composition of the plasma. Now, 45% of our blood, it is formed element of blood. Formed element. Less than these parts called Baffy code. Baffy
Go.
code. Baffy code includes W, BC, and platelets.
Platelet.
Yeah, it's a less than 1% Baffy code. WBC, you know, it's divided in granulocyte and
Agranulocytes.
Yeah, the normal number of WBC in 1 microliter, WBC in 1 microliter equal to 6,000 to 11,000. 11,000 in 1 microliter of blood. It's a normal number of WBC.
WBC.
If more than 11,000, it is leukocytosis.
Leukocytes.
Less than 6,000, it is
Leukemia.
leukopenia.
Leukopenia.
And then platelets, normal range of platelets, 150,000 to
300,000.
450,000.
450,000.
Yeah, it's normal. More than 450, thrombocytosis,
Cytosis.
less thrombopenia.
Thrombopenia.
Okay, about granulocytes, agranulocytes, you understand.
Yes.
Yeah, you know the most abundant cells of our WBC neutrophil, about 50 to 70% of the WBC, it is neutrophil, the most abundant and the most motile cell. Then, lymphocyte actually about 30%, 20 to
30%.
Then monocyte, 4 to 8%.
4 to 8%.
Yeah. Eosinophil about 1 to 4% or 2 to 4%. Basophil about 0.5 to 1%.
1%.
Yeah. This is the percentage of our WBC. Okay. The most abundant cells of our blood, it is RBC. Red blood cells. Red blood cell in 1 microliter equal to 5 to 6 billion. Billion. Yeah. Billion RBC present in 1 microliter. Do you know RBC or red blood cells? It is a biconcave cell.
Yes.
Yes, sir.
Biconcave cells, the diameter
7.5.
7.5 micrometer and thickness about 0.2 to 0.8.
8.
Okay? About hematopoiesis and so on. Now, about these cells, lymphoid progenitive cell. Lymphoid progenitive cells responsible for formation of our lymphocytes.
Lymphocytes.
That means lymphoid progenitive cells differentiate to the
Lymphocytes.
differentiate to the lymphoblast to the prolymphocyte to the lymphocyte.
Lymphocyte.
Lymphocytes divided in two types: T lymphocytes and
B lymphocytes.
B lymphocytes.
Lymphocytes, produce or formation of lymphocytes happen in the red bone marrow, but maturation of lymphocytes in other organ. Maturation of T lymphocyte happen in the
Thymus.
thymus. B lymphocytes in
Bone marrow.
lymph node, spleen. Okay, now about red bone marrow, you understand it is central hematopoietic organ. Second central hematopoietic organ, it is thymus. Actually, thymus, it is a central lymphopoietic organ, which is responsible for maturation, education, and training of the T lymphocytes. Yeah, T lymphocytes after formation left the bone marrow, circulate within the general circulation, and goes to the thymus. What is thymus? Thymus, it is a bilobar multilobular organ which situated anterior the heart in the medium mediastinum. Understand?
Yes.
Medium mediastinum, anterior the heart. It's a bilobar organ, multilobular organ. That means thymus surrounded by dense connective tissue capsule and divide by septa to the lobule like this. This is the thymus for example. This is a functional diagram. I want to draw for you. It's a bilobar organ. Yeah, consists of two lobes. Bilobar organ, which is surrounded by capsule and divided by septa to the thymic lobule. Here you can see the thymic lobule. Every thymic lobule consists of cortex, cortex. Here you can see the cortex and medulla.
Medulla.
Cortex is stained well because aggregation of T lymphocytes. Immature T lymphocytes aggregate in the cortex of thymic lobule. Yeah, and medulla of thymic lobule stain pale. Yeah, if I zoom one thymic lobule here, this is the one thymic lobule, which consists of cortex and this part is medulla. Here is outer part is cortex. Okay. In the thymic lobule present 6 type of thymic cell. Thymic cell. First type of thymic cells just situated beneath the capsule, also known as perivascular cell, responsible for formation of thymic blood barrier. Yeah, this cell. This is the first type of thymic cell. And in the cortex of thymus aggregate millions actually T lymphocytes. It means maturation, education, and training of T lymphocytes happen in the cortex of thymic lobule by thymic epithelial or reticular cells. Second type of thymic cell I draw here for you. Okay, this cells, thymic cells, which is responsible to secrete thymosin, thymopoietin, and thymic factor. chemicals. I draw here. I zoom 1 time X cells. Okay. This is 1 time X cells which produce chemicals: thymosin, thymopoietin, and thymic factor. And here is T lymphocyte. T lymphocytes immature. Yeah. Under the influence of the chemicals which produce this thymic cell, the T lymphocytes produce specific protein. This protein actually produced by rhabdomyoplasmic reticulum by ribosomes and then sent outside on the surface of the T lymphocytes, making this receptor which called CD, for example CD4 and
CD8.
another receptor here CD8
8.
receptor. Yeah, CD8 receptor. And also the last receptor which produce here or make, that is TCR receptor, T cell receptor.
T cells.
Yeah.
Not T cells.
And what do you think? This thymic cell responsible for education or stimulation of these T cells. These T cells, immature T cells, should make this CD4 and CD8 and TCR receptor in their surface. Now, these T cells mature and should move at the first toward the medulla. In the medulla present blood vessel and left the thymic globule. Okay. And should pass the positive and negative selection. Do you know what is the positive selection? It's a checkpoint. It's a checkpoint, yeah. For example, student complete the first semester, yeah?
Yes.
And should pass the first step of final exam and second step. First step it is positive selection and second step it is negative
selection.
Yeah, that's a checkpoint. Okay, here another cells present, also thymic reticular cells type 3 here present, which has specific receptor. This receptor called MHC2 and MHC1. MHC2. Okay, this thymic cells responsible to check CD4 and CD8. Yeah, if CD4 and CD8 receptor recognize the MHC1 and MHC2 receptor and fit each other, it means these T lymphocytes normal. Qualify the positive selection. Positive, it means they should recognize each other. Yeah. Recognize. This is the positive selection. Unfortunately, about 85% of T lymphocytes can't pass or can't qualify the positive selection. It means CD4 and CD8 receptor not recognize the MHC1 and MHC2 receptor. If not recognized, these cells produce very specific enzyme which is called phase enzyme. And phase enzyme kill the T cell. And also in the cortex here present macrophage which is responsible to engulf or phagocyte the corpus of dead lymphocyte. Clear?
Yes.
If 85% T lymphocyte after maturation not qualify the positive selection, it means only 15%
Qualified.
and they survive and move toward the medulla. In the medulla present another cells, another thymic cells which known as dendritic cell. Star shape. Yeah, dendritic cell. This dendritic cell, star shaped cell, this is the thymic dendritic cell. Check, make second checker. It means check the TCA receptor. T cell receptor. How check? Do you know this TCA receptor is very significant receptor because by this receptor T lymphocytes recognize the antigen. Recognize
Antigens.
Yeah. How you think? How dendritic cells check the TCA receptor?
Antigens are present.
Yeah. Take own tissue of a peptide, a molecule peptide of the own tissue present for the TCA receptor. For example, give this, take this marker, break it down. It means you not survive. You should die. Because what? Because you not should take this marker and break down. Because we work with this. Yeah? This dendritic cells also represent for this cell on tissue. If this cell recognize on tissue, not catch the molecule, and if not catch, it is negative selection. That's why I call it. It means recognize on tissue. If catch and break, means it's abnormal T cells which not able to recognize on tissue and they should die.
Yes.
Yeah? Normal, like this, dendritic cells check the TCR receptor. It means take the present, this dendritic cells present on antigen for TC receptor. TC receptor, if recognize on antigen, reject. If not recognize, catch the antigen and
Break it down.
Breakdown.
Yeah, damage.
It breaks and it's
Yeah.
negative.
If reject, it means negative
Negative.
selection. It means this cell, T cell, survive
Hmm.
and they not kill because understand it is on tissue. If catch, it is this T cells abnormal and should die.
When it's catching and then it's breaking, what is the negative or positive?
It's, do you know if reject the own molecule, it is negative.
Negative.
Normal situation should reject.
Reject.
Yeah, that was called negative. If catch, it means it's abnormal.
Abnormal.
Yeah, positive.
Positive.
In the medulla, positive selection not happen, negative selection. Okay, now, after only 2-3% of the T lymphocytes qualify the first and second selection, positive and negative.
Negative selection.
Only 2-3%. And then let the time the globe will circulate within the general circulation. Yeah. For example, you are T lymphocytes which came from India, and this university is Thymus,
Thymus.
responsible for your education, training. Yeah. And the teacher of university is thymic cell. But you are not like thymic cells. Yeah, if
Rejected.
you will not qualify the positive and negative selection, if you not even qualify the exam, you go back. Yeah.
Fail.
And thymic cells directly if they not qualify. Yeah. Okay, 2-3% of the T cells, 2-3% of the T cell only able to pass the positive and negative selection and leave the thymic globule and circulate within the general circulation.
Circulation.
Okay. What is the function of T cell. Who can tell me?
Immune T cell. Immune.
Do you know how working our immune system? For example, this, what is this structure? How you take it? This structure called microbe, for example. There's a bacteria. Yeah, if you eat something, yeah, with bacteria, this bacteria in your GIT system, in the mucosal layer of GIT system, enter in your tissue. This bacteria very intelligent, rapidly, quickly multiplying. Yeah, they multiply, increase in number, and at the same time they release exotoxins and antigen to damage our tissue. Yeah. In our tissue present another very beautiful cell. In everywhere present this cell. This cell called mast cell.
Mast cell.
Yeah. Mast cells produce chemokines,
Chemokines.
interleukins, for example, and it means send signal for other our white blood cells. Send signal for our white blood cells. White blood cells should move purposefully toward the microbe or bacteria and fight with them. The first white blood cells which fight with microbe is neutrophil, because neutrophil is the most abundant and the most motile.
Motile.
Yeah, neutrophil is also called microphage and antigen presenting cell. Yeah, neutrophil move here. Yeah, to fight with microbe. And other white blood cells like monocytes also come here to fight with microbe. Monocytes macrophage, neutrophil microphage, but they are both antigen presenting cells. Yes, these two cells at the same time, which they fight with microbe, they release an interleukin, interleukin 1, interleukin 3, interleukins, and also colony stimulating factor send signal for our red bone marrow to produce more white blood cells, and also send signal for other white blood cells to move, fight with microbe. Okay, neutrophil and monocyte fight with microbe. For example, this monocytes phagocyte the bacteria. Phagocyte and digest the bacteria and take the antigen, a very small part of the cell wall of the take on their surface here. This is the antigen, for example, which this is the antigen of bacteria. These two cells, how take the antigen of bacteria, move to the T cell. Here, find the T cell. Present this antigen for T cell. T cells as T cells are particularly TSR receptor, and T cell catch this antigen by this TCR receptor catch and analyze the antigen, and T cells goes to find the B cell, B lymphocyte. Yeah, B lymphocyte, and stimulate or convert B lymphocytes to the plasma cell. This is the plasma cell.
Plasma cell.
Yeah. What is the function of plasma cell? Plasma cell produce, what is this? Antibody.
Antibody.
Yeah, this is the antibody. Million of the antibody produced by plasma cells, because plasma cells in the cytoplasm contain the rapid plasma reticulum, Golgi apparatus, and also the ribosomes actively, rapidly, quickly produce, continuously produce antibodies against this antigen which gave T lymphocytes for the disease. B lymphocytes convert to the plasma cells release the antibody. Antibodies circulate within our general circulation. We will find the antigen of the microbe block or neutralize. For example, this is the antigen of microbe, everywhere damaged our tissue. This is the antibody. Yeah, every antigen have special own antibody. Yeah, which made by plasma cell. And neutralize the antigen. Now this antigen not damage our tissue. Yeah, understand?
Yes, sir.
How working our immune system? White blood cells responsible to fight with microbes, phagocyte the microbes, and present the antigen of microbes for T cells. T cells activate or convert B cells to the plasma cells. Plasma cells produce antibodies neutralize the antigen of microbes. Clear?
Yes.
sir.
Okay, what you should do today? You should draw the specimen of red bone marrow and specimen of thymos. Yes, of course.